Thank you. And first of all, I want to thank Bill for this incredible honor that I've been invited and to give a talk here. And I want to thank all of you for participating. And it's kind of interesting. I'm a physician scientist again. I see kidney cancer patients. And I'm very grateful that I can see all of you. I normally talk to either scientists or physician or pathologist. I never kind of have this big audience or data scientists. But I think this is very, very exciting to me because when everything started about 25 years ago when I was a medical student. In that time, cancer patient when they were diagnosed, there was no treatment. And that's why I came to Hopkins to study molecular biology. And at the end of my career of my PhD, I did very well, but I felt like there was no way I can ever translate the data or the knowledge I obtained from molecular biology to help patients. And that's why I went back to finish my medical training. And I trained as a medical oncologist, and I did biochemistry, mouse models, and I went back to WashU to be a faculty and to see patients. And then when I was at Harvard Medical School, I was seeing lung cancer patients. But when I went back, they say, well, why don't you see kidney cancer? And that's how I started to see kidney cancer patients. And there was a time that I call it dark age, because at the time there was only 2 drugs, and then most of the patients die of the disease within 15 months. There's no good treatment. And at the time, we still have no idea how we're going to treat the disease. But I think from 2010 and onwards, there are next generation sequencing and everything ongoing. So I think we are getting into a time that we have so much data and we have so much knowledge, and then we can actually make a difference. And there are so many treatment options available. And I think that the next thing we'll try to do is really try to kind of personalize all the treatment and then make it cheaper and more effective. So that's why I think that's why you're here. So you want to cure cancer. I will give you a few. This is very, very simple thing to think about it. So cancer is like this bird living in a very good, well protected environment. So if you just want to shoot the bird with something that's ineffective, it's not going to work. So you better have something to cut out the wood, and the wood is the microenvironment. Okay, so the cancer is not lived by themselves, live with the environment. And then you can, with removal of the environment, then you can have a better shot at the cancer. And now, of course, of all those, if they all go crazy, you will bring the drone, and it's the immune system, your own immune system, and kind of to try to destroy it. So that's all you need to know how to cure cancer. Okay, I think after this, everybody know how to do it. So again, the key is really teamwork. And I think you guys here is really represent the mad scientist part of the Albert Einstein. So we have some of the surgeons, some of the oncologists, the radiologists who make all the diagnosis and so on and so forth. Then we can find the drugs and find out the target and how to treat the patient through a clinical trial. So there are many ways of working together to help. And so the scientist part become very, very important is because all the others are sort of, I feel like so all established for the past century, 2 centuries already. But the scientist part is really how things are going to move forward. And just to, we are going to talk about rare kidney cancer, but I think to me, cancer is basically, to me, it's very, very similar. It's although every cancer is very different, everybody's cancer is different, even if individual patients' cancer, they are very, very different, but they all follow the same idea and to move forward to develop, cause a cancer. So what we're trying to say here is a kidney cancer. So it's not a very common cancer type. It has 60,000 new cases and quoting 14,000 fatality in the United States. So when the cancer in the solid tumor, when there is localized, the surgery most of the time is the way to provide cure. And then if you have an advanced disease, then you have to start thinking about chemotherapy, radiation therapy, and so on and so forth. So kidney cancer is kind of interesting in a sense. There's a disease, cancer in the kidney, and originated from the kidney, but it's not only one disease. Because it's actually have more than 10 different subtypes in the kidney. And then the most common type is called clear cell type kidney cancer. And the rare types are the others we call rare type, we call it non-clear cell. And Bill's tumor is the papillary type 1. It's 5 to 10% of kidney cancer. It's not as common. And that's why we don't have any a very good idea about how to treat them and how to kind of manage their disease once they develop metastasis. So I put up this slide. It's kind of interesting. I started seeing the kidney cancer patient. I call it dark age. It's before 2004. There was only 2 drugs available. Those are immune-enhancing cytokines. But the following 10 years, I call it a modern age because we have targeted therapy. There are 2 different colors that represent 2 different treatment options. And then basically, that single-arm targeted therapy actually moved the curve from 15 months of median survival to 30 months. And now recently, from the 2015 and onward, we have 4 different kind of treatment available for kidney cancer patients. And then I call it golden age. And I think with all those available treatment plus precision medicine, combination sequencing, the treatment and immunization, prevention, detection, we should be able to. So that's why I was talking about, the very beginning is take kidney cancer as an example from dark age to modern age. Currently is golden age, and I think that for the next 10 years or so, I hope that we can move to diamond age. And this is the same concept to be able to apply to any cancer type, not really just kidney cancer. So the problem with rare disease is that common disease is we spend more efforts. We're easier to understand because of the material, and so you can see from the left-hand side the clear cell kidney cancer. The survival has changed dramatically, but all the other non-clear cell cancer patients, they didn't change much. So that's why it's become very important to understand how to treat in the rare disease, especially like rare kidney cancer. So I think what we need to do is we need to understand the biology. We need to learn how to classify them, and the teamwork. And novel approaches. And novel approaches basically the scientist's work. And that's why I think I'm excited to talk to you all about this opportunity. And that's why we want to inspire some of you to think about how cancer actually evolved. And so instead of talking to you the very, very detailed network of how cancer is formed, and I try to come up with some simple concept for you to kind of digest, to understand, and think about it, how human cancer is built. It's complex, but we all have 26 chromosomes, 6 billion base pair from a single cell, and develop a human with 2 eyes, one nose, one mouth. So it's the same thing. The cancer is using the same genome to build cancer, but it's just a much rapid evolution, and then it's just causing more problem, and they actually kill itself. So in a human, on average of us, about maybe 50 trillion cells. As I say, it's a 6 billion base pair per cell, and that represents about 20,000 genes per cell. So the problem is that at this moment, you have to think about is that the 50 trillion cells and 6 billion base pair per cell, we have to repair and replenish about 100 billion cells a day. So that means you have 10 to 21st order of magnitude DNA base pair transaction in your body every day. And that's why you have to have error to occur. If you have zero error, then there will be no evolution. But if you have too many errors, you are going to cause problem. So basically, the cancer basically you start with a genotype and then goes to the phenotype. So you start with DNA, and then you have the epigenome, there's RNA, the DNA transcribed to RNA, RNA translated to protein, and protein media all the metabolic transaction. That's why we have this daily activity. And so DCGA on kidney cancer spent a lot of efforts for the past couple of years. We actually participated in to further characterize all these cancer types. And with the platform that you have now, have DNA, and methylation is for the epigenome, microRNA for the epigenome, and microRNA, messenger RNA protein. The only thing that you don't have is metabolites. But these are all essential platforms that you are going to encounter nowadays with everybody. But the issue we actually be able to kind of understand the disease much better because with the sequencing availability that we now know the certain genes, certain disease like kidney cancer. In the past, we only think maybe there was one gene, but now we actually figure out there are many genes are involved, and we will be able to understand the disease biology much better now compared to a couple of years ago. So the other major problem that people will start to encounter and seeing is that the heterogeneity. We are not talking about the heterogeneity between patients. We are talking about heterogeneity within the one single tumor, within the one single patient. So if you have a big tumor, basically have billions of cells and started with one cell, but they accumulate different kind of mutation and they grow stronger, and it's easily represented 1,000 different subclones, and they have some different ability to carry out some actions. So this paper published by my friend, a colleague, and Charlie, and a couple of years ago, basically changed the way we think about cancer. Basically, we're thinking, oh well, treating with kidney cancer, everybody's kidney cancer is the same. No, everybody's kidney cancer is different. Even the kidney cancer within the same patient is very different. So that's the major challenge that we are having. The sequencing, you are sequencing a bulk of the tumor, but you are not actually looking at the individual cells of them. So there are many heterogeneity available there. So it's like, so the idea was proposed that it's like a tree, it keep branching out. So almost there's no really any hope that we can actually do anything about it. But we took a different approach about the same time, that we look at the patient that actually respond to very single agent targeted therapy very well, and we do the similar multi-regional sequencing and try to understand why this patient, if tumor evolution is so random, so how can you actually using one drug to treat it? So when we do multi-regional sequencing, what we found out is yes, they have heterogeneity, but they actually converge on similar pathway or similar genes, and it's a convergent evolution. So that actually starting for the very first time, I think that the cancer evolution is tree in a sense from a very detailed perspective. But from the functional perspective, a pathway perspective, they actually converge on certain pathway to activate certain things, to carry out and obtain specific capacity. So for example, that they want to pick up different mutation, they want to gain different capacity to carry all the cancer hormones, because they have to proliferate indefinitely, they have to evade immune surveillance, and they prevent and avoid cell death, and so on and so forth. So a couple of years ago, that's why the first time I was talking to Bill, I think, so the cancer evolution is sort of like a braided river. It branch out, but it can converge on something. It's restricted by lots of different things, from the tissue restriction, or you have immune restriction, or and so on and so forth. So it's not as random as we thought. That's totally just went crazy. But it can go crazy when I was telling you at the end that they have already obtained all the capacity they need to become a cancer, and then we pick up additional driver that I call a speedy driver. By that time, basically, they have no rules. And by the very beginning, they have rules. If they want to develop, they have to have rules. So they try to gather a different capacity, different phenotype, different function to build a cancer. So the very beginning is restricted. So you want to treat it, and especially when you start to the river start to evolve, you can actually try to understand what kind of river this kind of disease is going through. And before that, you can think about what kind of the backup pathway, the bypass pathway may be there, and then you can treat them preemptively. By treating them with multiple, the targeted therapy failed is because in the past people just gave them one drug. And now you know that it's going to have bypass pathway, and the compensatory pathway is going to happen. And that's why it failed. So if you can predict, if we can predict, and I think we can, and I think we have the capacity, I think we have the data, we just need to know how to take a look at it and to come up with a very smart idea about what kind of compensatory mechanism may be happening in this specific disease, this specific patient, based on their limited mutation profile, based on their early mutation profile. Then we can actually treat it better. So I want to, so the thing is basically it's all concept driven. So I give you the idea about in another tree. The tree is kind of, it's kind of interesting, easy to explain everything, but it's hopeless to me, and we cannot treat it. But the river actually tell you a little bit something that maybe there's some, bottleneck. You can take care of it. But then we recently, based on the genomic data, we generate mouse model, and I'm going to tell you some concept that Ben was referring to that I just came up a couple of days, maybe a week or two ago. But I think I was kind of going to share with you because I think it's kind of interesting concept. So I call it a oncogene tumor suppressor gene circuit breaker hypothesis. So this is taking, so 20,000 genes in our body. Some of them are pro-proliferation, and then if they go crazy, they cause tumor. That's why it's called oncogene. And some of the genes in our body is to control those genes not go crazy, and it's called tumor suppressor genes. But those genes are not there to be oncogene or be tumor suppressor. They are there to protect us. So they are, the oncogene and tumor suppressor gene, they are actually forming pairs. And I think this one is one that I want to tell you and kind of build step by step and telling you why I think the oncogene and tumor suppressor circuit breaker is actually operating in all our body to help prevent us from getting cancer. And you can reactivate the circuit breaker and repair the circuit breaker and to treat cancer cells. Okay, so I want to give you the example. It's very clear from our mouse model, from everything that could show it here. So there are two genes that you need to know. It's called VHL, and when it's lost, causes clear cell kidney cancer. And it's almost all the clear cell kidney cancer have that gene loss. And there's another gene, it's called hypoxia-inducible factor. So when you have tissue injury, this gene gets stabilized because the tissue, they have low oxygen. And this HIF is a DNA protein gets stabilized because low oxygen. Because it's oxygen sending mechanism in our body. So because of low oxygen, HIF gets stabilized and they stimulate blood vessel formation. Okay, so that's our repair. If you don't have that, you cannot repair. You'll bleed to death. You cannot repair yourself, right? So that's almost all oncogene is doing, is that to help us to reach homeostasis. Okay, so that's what is going on. That's why when you have hypoxia, the output is just the line that I'm showing you. So the next thing what happen is that if you have VHL loss, what happens is that HIF activity not only controlled by oxygen, is really controlled. It's become act up because it is an active control. The first circuit breaker is broken, so the HIF is activated. But HIF activated, it activates another circuit breaker to break it, to make sure it doesn't go just one single mutation cause tumor. Okay, if everything is one single mutation, one circuit breaker broken cause tumor, we have tumor all the time. Okay, so there are many multiple fail-safe mechanisms in our body to break it. And so from VHL mutation, the oncogene get activated is HIF. And when HIFs get activated, there's only a few circuit breakers there to break HIF's function. Okay, and this circuit breaker that I'm telling you is the most prevalent in a clear cell kidney cancer is called p1RCC. So that protein basically is another circuit breaker to prevent HIF overdrive. Okay, and when that circuit breaker is broken, so then HIF signal further upregulated and also collaborate with the STAT. And those genes actually trigger the more intense output of the HIF output and STAT output. And what happens is that it still doesn't cause tumor, it still needs to trigger it because it activates another circuit breaker, and the other circuit breaker needs to be broken again before the tumor can occur. Okay, so the third circuit breaker is the TSC, and tumor suppressor mTOR is a gene that helps us manage our nutrition. So these are many circuit breakers, and you can think about it. Once the tissue type specific circuit breaker broken, they have to activate different kind of oncogene. And when the oncogene is activated, it activates some tumor suppressor activity, and that is it because the oncogene associated tumor suppressor activity is really they activate the inherent circuit breaker. And the other thing is kind of interesting. So all the study you can see here is the other gene is called STAT, and STAT is kind of interesting. It's very important immune regulator transcription factor. But when the p1RCC is lost, the STAT and work with HIF to cause tumor genesis. But interestingly, STAT is a very important immune regulator. And then, interestingly, is that when these three circuit breakers broke, then you can cause tumor because you need a HIF pathway, you need mTOR pathway to be activated. I put this another circuit breaker there. It's called immunosurveillance. In our body, our immune system actually doing a very, very good job to pick out the tumor cells. Not only the repair mechanism, not only so the immune system actually is doing a very, very good job. And if our immune system is not doing a good job, I think we get tumor all the time. And I always tell people that I think I have tumor every time, every day. Because some of them, I just didn't reach the time, to reach the stage to manifest. And the immune system actually doing a very, very reasonable job to us. So I think the new concept of cancer cure is really, cancer can be disease-free, no disease coming back within a couple of years, and you don't see anything. With not any treatment, they should be sort of cure. But the cure is sort of philosophical, because I think I have cancer all the time. Okay. So the other thing that I want to talk about it's kind of interesting. The immunotherapy has become very, very, promising, and has been shown to kind of actively helping a patient like kidney cancer, lung cancer, melanoma, bladder cancer, many other cancer types. And it is kind of interesting. And the most interesting thing is that when people try to do biomarkers, trying to figure out what kind of kidney cancer patient will benefit from this immunotherapy, they found out PBRM1, the second circuit breaker loss, actually predisposed patient and kind of predict patient will respond to immunotherapy. And the reason, based on our model, you can think here is that if when the PBRM1 is lost, HIF1 is activated, STAT is activated, so the immune system is actually ramped up. But so there's another circuit breaker there is to prevent cancer from happening. But it's broken again through the PD-1 expression, because the circuit breaker is kind of broken. And then the thing that they actually work for immunotherapy is the anti-PD-L1 antibody and things like that. So basically reactivate the lost immunosurveillance circuit breaker. So I think that's sort of a concept that you make it to think about it, not looking at oncogene perspective, but look at a tumor suppressor gene perspective. But tumor suppressor gene not really through mutation, because it could be through silencing of the transcription, inactivation through many different ways. So the oncogene and tumor suppressor gene form the circuit breaker. It's likely context dependent, meaning like the tissue dependent, because tissue have restricted them from causing a tumor. And the last, this is the papillary type 1 kidney cancer. And the DCGA has spent a lot of efforts to characterize those. And the most important thing that we know is it's almost like chromosome 7 gain is almost always happening. So this suggests this is the very, very initial event to cause tumor, not necessarily through mutation, but through copy number changes. Just like clear cell kidney tumor, the first event is not, the VHL is lost in 90% of patients, but the first event is chromosome 3 and chromosome 5 through some event called chromothripsis, and lost chromosome 3 and gain chromosome 5. And so there are lots of time. So in the past, when we don't have sequencing capacity, people look at copy number and they think, oh, this is all copy number coding problem. And people now can do sequencing, they say, oh, it's all mutation coding problem. But it's actually at least 50-50. Okay, so you cannot just look at mutation and you'll miss the big boat. And the other thing you have to look at, make sure you understand which is a very early event, so it's a truncal event, it's a highly prevalent. What are the other tertiary event? What's the speedy driver? They will be less frequent. And here is basically telling you again. So what we have learned from papillary type 1 is a chromosome 7 gain, and it's Y7 gain. It's because MET gene is also mutated, and MET gene is on chromosome 7. So it's actually telling you that could be a very, very fundamental driver at the very beginning for this disease. Of course, there could be something else, but that's why we need to spend more effort trying to figure it out. And here is basically just to tell you that all the data sets are available. They have protein data, they have microRNA, they have copy number, the messenger RNA, DNA methylation, and those kind of things. They can help you to further subcategorize patient. But again, this is a bulk, right? So every patient is one line. But to me, each patient actually has maybe 5 subtypes. So what you need to figure out is how to preemptively treat those 5 subtypes from that patient, not just treat one. Okay. So anyway, so that's another term I created. It's called integrated cancer therapeutic cycle. So we started with biomarker, we had study mechanism, animal models, therapeutics, and the omics is the thing that actually changed everything, I think. And the data, because when I studied was a graduate student, that was the very beginning of internet. Now everything is so we cannot live without a phone, like watching it. I was like, it's kind of crazy, but that's the world now. So, but it also gives us the power to analyze things, to integrate things. So the key is really how to analyze and integrate things. I think this is really related to the keys and locks ideas. So every is a puzzle. Every cancer case, every rare disease is a puzzle. And the key is you use your brain to get all the data to find out the key to solve the problem. So what I want to do is to help all of you, and I hope that all of us can continue to explore and try to find the way to get rid of the rare or more cancer. The other thing I want to tell you is this. We will all get cancer. Cancer is an aging disease. If you can live longer, okay, your DNA continue to repair is 10 to the 21st order magnitude of DNA keep so your body is basically a registry of mutation, and something is going to happen. Okay, but I think the most important thing is to live a healthy life, happy life, not forever. Okay.